Welcome to this explainer. Today, we're diving right into the absolute most fundamental molecular building blocks of life, and we're going to see exactly how they're powering the next generation of medical miracles. Yep, we're talking about DNA and RNA. So whether you're a biology buff or just, fascinated by how fast modern medicine is advancing, you are definitely in the right place. Let's get into it.
Think about this for a second. What if we could rewrite the actual software of life? It's a huge question, right? Because understanding how to treat those conditions we've always thought of as untreatable starts right here, at the fundamental code inside every single one of our cells. I mean, to fix a computer glitch, you got to know how the software is written, and it turns out the exact same logic applies to the human body.
And this right here perfectly illustrates that code. It's that beautiful, iconic, twisted ladder of the DNA double helix. This elegant structure holds all the strict biological instructions needed for any living thing to grow, develop, and reproduce. It's quite literally the physical manifestation of our genetic identity, just passed down from generation to generation. So here's our roadmap for today's explainer. We'll start with a master blueprint. Then look at the structure of the helix, meet the messenger, compare DNA versus RNA, and finally, wrap up with the future of therapeutics.
All right, part one, the master blueprint. Let's talk about DNA as the absolute foundation.
So DNA, or deoxyribonucleic acid, if we're being formal, is basically the ultimate hard drive of your cells. It safely stores all that long-term genetic info that makes you, well, you. It hangs out safely inside the nucleus of eukaryotic cells, with a tiny bit also found in our mitochondria, and it codes the information for literally all biological processes: metabolism, growth, reproduction, the whole shebang. It's this massive self-replicating biopolymer, basically a giant biological chain. But what is that chain actually made of?
Well, to really understand this master blueprint, we have to zoom way in and look at its building materials at the atomic level. DNA is made of these repeating units called nucleotides. And to grasp the whole picture, we really need to look closely at these individual little pieces.
It's crazy to think about, but every single nucleotide is built from just three basic components. You've got a phosphate group, a sugar, specifically deoxyribose, which just means it's missing one oxygen atom, and a nitrogenous base. These three parts link up to form the incredibly long chains that make up our entire genetic makeup.
Now, those nitrogenous bases are where the magic happens, because they're the parts that actually carry the specific code. They fall into two structural camps: the single-ringed pyrimidines and the double-ringed purines, like adenine and guanine. The specific sequence of these bases acts as the biological letters of our genetic code. That sequence is exactly what determines your unique traits.
Moving on to part two, structure of the helix. Let's see how this ladder is built.
So think of DNA like a twisted ladder. The sugar and phosphate units link together using these incredibly strong covalent phosphodiester bonds. It's essentially unbreakable chemical glue that creates the sturdy handrails of the ladder. But here's the really fascinating part. Those two outer strands, they run anti-parallel to each other. So one strand runs in what we call a 5' to 3' direction, and the other runs in reverse, 3' to 5'. And that opposite polarity is an absolute game changer for the stability of the whole molecule.
And that brings us to Chargaff's rule, which is absolutely crucial. It dictates exactly how the rungs of our ladder are formed. See, these bases don't just randomly stick together. No way. Adenine always pairs with thymine, connected by 2 hydrogen bonds. And guanine always pairs with cytosine, using 3 hydrogen bonds. Now, individually, hydrogen bonds are pretty weak, but when you add them up across millions of base pairs, they are incredibly strong, holding those 2 complementary strands together perfectly.
Now, DNA can actually contort into a few different shapes, depending on things like salt or humidity, like the A form or the left-handed Z form. But the primary shape existing inside your cells right this very second is known as bDNA. It was originally described by Watson and Crick, and it's a right-handed helix with 10.5 base pairs per turn. It's the standard form under normal conditions. And notice those alternating major and minor grooves? Those asymmetrical gaps are super important because they allow proteins to easily get in there and interact with the DNA.
All right, part three, meet the messenger. Let's introduce RNA.
Okay, so we've got our highly stable blueprint, and it's securely locked away in the nucleus vault. But, a blueprint can't build a house all by itself. Enter RNA. If DNA is the master architect, RNA is the active worker on the ground. It literally photocopies the DNA's instructions in a process called transcription. Then it travels out of the nucleus, into the cytoplasm, and heads straight to the ribosomes. Once it's there, it actively translates that genetic code to build the actual proteins that keep us alive and functioning.
And unlike DNA, which really just has that singular role as a storage vault, RNA is incredibly versatile. I mean, only certain genes are expressed into RNA, and they take on all sorts of specialized forms. It's like a tiny biological factory. You got mRNA carrying the actual work orders, tRNA acting like little delivery trucks bringing in amino acids, and rRNA actually making up the factory machinery itself. That's an entire highly specialized fleet, just to ensure our proteins are built perfectly.
Which brings us to part 4, DNA versus RNA. Time for the ultimate showdown.
This breakdown beautifully highlights their chemical differences. Let's start with structure. DNA is this long, double-stranded chain, while RNA is a much shorter, single-stranded molecule. Now look at the bases. They both use adenine, guanine, and cytosine, but notice how RNA swaps out thymine for uracil. And critically, look right there at the sugar. DNA uses deoxyribose, but RNA uses ribose. RNA actually has an extra oxygen-containing hydroxyl group.
And honestly, that one tiny chemical difference, that extra oxygen in RNA, changes literally everything. It makes RNA far more reactive and short-lived than DNA. DNA is highly stable and it self-replicates, which makes it the absolute perfect material for long-term storage. RNA, on the other hand, doesn't self-replicate at all. It's synthesized on demand and then degrades pretty quickly. So their chemical structures perfectly dictate their different jobs in our genetics. Finally, part 5. The future of therapeutics, how we're hacking human disease.
So why does this microscopic chemical difference matter to you and me? Well, because it is the absolute key to hacking human disease. The biotechnology field is experiencing a massive boom right now because we're using these distinct properties to build truly revolutionary medical treatments. We're basically treating DNA and RNA like two completely different software patches for the human body. DNA therapeutics aim to actually replace or repair defective genes, whereas RNA therapeutics, they simply tweak or modulate protein expression without touching the core code.
Let's look a bit closer at DNA therapies. Since DNA is meant for long-term storage, these therapies are aiming for a permanent fix. They're incredible for inherited monogenic disorders, diseases caused by just a single defective gene, like certain types of hemophilia or retinal dystrophies. By delivering a corrected copy of a gene, usually using viral vectors, we can potentially cure the disease forever. But because it requires integrating that new DNA directly into the host's genome, it definitely carries risks. Things like insertional mutagenesis, which is when the new DNA accidentally disrupts a healthy gene. So, delivering it safely is a massive, complex challenge.
On the flip side, we have RNA therapies. Remember how we said RNA is transient and highly reactive? Well, that makes it amazing for swift, reversible treatments. Delivered safely into our cells via these cutting-edge lipid nanoparticles, RNA molecules don't ever integrate into your permanent genome, so the mutation risks are significantly lower. They just drop off a temporary message. This tech is absolutely ideal for complex diseases, cancers, and, as we all lived through recently, it's what powered the massive global mRNA vaccine response to COVID-19. By really deeply understanding the unique molecular strengths of both the stable long-term blueprint and the reactive temporary messenger, science is unlocking incredibly precise, tailored treatments for an unbelievable array of diseases. It's not just a matter of picking one or the other anymore. The complementary use of both is literally changing healthcare forever.
I'll leave you with this final thought. We are standing at the absolute frontier of biotechnology right now. With the rapid pace of personalized medicine, we aren't just quietly reading the software of life anymore, we're actively writing it. So, as we continue to master these molecular languages, what previously untreatable condition do you think we're going to cure next? Thanks so much for joining me on this explainer, and hey, stay curious.